Lovely. Thank you. Ladies and gentlemen, thank you so much for coming back so promptly. We'd like to continue this afternoon with our panel session and hopefully get you out of here on time. There will be an opportunity for questions as well. So it's with great pleasure that I would like to introduce the chair for this afternoon's panel discussion, Professor Elizabeth Coulson, who is the group leader of the Clem Jones Centre for Ageing, Dementia and Research. Elizabeth started at QBI in 2002, so she was here for the actual start. And she is also co-chair of Dementia Australia's scientific panel. So please, a warm welcome for Professor Coulson. Thank you.
Great, and welcome back, everyone. I hope you got a little bit to eat. What I'd like to do is just do a little bit of an overview of what we're going to hear today. So at QBI and in the Clem Jones Centre, much of what we do is very much at the fundamental level, trying to understand the mechanisms that happen in dementia. How can we think about curing a disease such as Alzheimer's disease or frontotemporal dementia if we don't understand what's actually going on in that disease? How can we possibly try and restore a memory if we don't yet understand what a memory is? And so that's what our panel members are going to tell you about today. But just to set the scene, so we've heard a bit today about plaques in Alzheimer's disease, and here's just a section through a piece of a brain. This is a mouse brain. If you remember the picture that Jürgen showed, it's really tiny. And this is just a small section of it here, and you probably can't see, there's some purple dots and there's some pink dots, and here's just a region that's blown up even more, these purple dots, and they're the cell bodies of the nerve cells or the neurons, and this pink blob here is a plaque, the cluster of beta amyloid that's aggregated together and deposited in the brain. And you can see along this line here, there's a gap around the area where the plaque is because those nerve cells have died. Okay, so what we're really talking about when we talk about dementia is a neurodegenerative disease, and we're trying to understand how degenerative processes occur. So something that Jürgen's already talked about is that there are many diseases, many dementias and many neurodegenerative diseases. And this is just one way they can be looked at. So this is Alzheimer's disease here. And the pathology in Alzheimer's disease is the amyloid plaques and the tau tangles. But as Jürgen has said, there's overlap with Parkinson's with dementia or Lewy body disease, Parkinsonism, frontotemporal dementia, and you can see that they all overlap each other. And all of these different diseases have a different form of an aggregated protein. And so one of the things that we're doing as scientists is trying to understand how these aggregated proteins or their earlier forms, the prototypical that aren't deposited in plaques yet, but the just few aggregates are causing toxicity to our neurons, our nerve cells, to try and understand the generic processes that are going on. We know in dementia that we start off cognitively healthy, and we can now look in the brains of people who are alive, and we know already that about 30% of people who are cognitively healthy have levels of beta amyloid in their brains that would put them diagnostically into an Alzheimer's category, but they're still cognitively okay. Then we have mild cognitive impairment, and at this stage there's already obvious neurodegeneration when you look on a brain scan at the structure of the brain. There's already degeneration occurred. But it's when you get a diagnosis of Alzheimer's disease, or in fact any dementia, that there's already these pathological aggregates in the brain and synaptic and neural degeneration, and these are all underpinning the cognitive changes, okay? But the point here is that there's already changes happening quite early on, and so in trying to treat a dementia, we really have to be treating here at the border between cognitively healthy and mild cognitive impairment. And to be able to do that, we need to understand the mechanisms that occur early in dementia, to target them with new treatments that treat the changes that are happening early in the disease process, to stop the disease process occurring. So what we're going to be talking about with our panel members is we're going to delve down through the brain to the neurons, the nerve cells. And this is a picture you'll see a bit later on in one of the talks. So nerve cells or neurons are one of the most predominant forms of cells in the brain. For the most part, you're born with the neurons you have, and they don't, you can't replace them. There's some very select parts of the brain where they do get replaced, but for the most part, the neurons in your brains don't get replaced. So here is the cell body, the active part of the cell. This is where the nucleus, where the DNA is, and this is where, for the most part, the molecules that keep the nerve cell active are generated. They get transported along their axon down to the end here, and there's also these dendrites that is where the information comes in. So there's the soma, or the cell body, the dendrites, and the axon, okay? So this is a stylized diagram of neurons, but to see what neurons really look like in the brain, there's a very famous thing called Brainbow, which labels all the neurons in, again, a mouse brain. And this is what they can look like. They're all labelled in different colours, so you can pick them out. This is not every neuron in the cortex, just probably about 30% of them are labelled, but actually you can see here is the cell body, and you can see their long processes up here and the dendrites down here. So this is really what we're talking about. So the dendrites is where the incoming information is, and which is coming in here, and the axon terminals here is where the information flows out, and that will be important. So what happens in dementia is that there's degeneration of the neurons, and this means that the communication between the nerve cells doesn't happen properly, and therefore our cognitive function can't happen properly, okay? And this can happen for a number of reasons, but one of those is that actually there are build-up of things like beta amyloid or tau that interferes with those processes, either by physically blocking the communication, causing degeneration, or changing the intracellular structure. So the communication from this cell to this cell, and this cell to this cell. I'm not sure what's... Sorry, we'll just jump on. So one of the things we often do in the lab is, rather than studying every single process at once, we try and narrow it down to be very reductionist, so we can study one thing at a time. And for example, looking at neuronal toxicity, we can study what amyloid beta aggregates do to cells, and we know that they're toxic to neurons. And what we're going to hear about today in 3 talks is, firstly, the toxicity can cause generation of free radicals, or reactive oxygen species, that we know is very damaging. It can damage the proteins in the membranes and cause neurodegeneration. We're also going to look at spine degeneration. So these are specialised function protrusions on the dendrites and the axons, and the compensation that might happen with degeneration. And finally, we're going to look at changes to gene expression in the situation of Alzheimer's disease where there's amyloid aggregates. And I think that's all I'm going to talk about today. So that's just to give you an overview. So today we're going to hear first from Professor Massimo Hilliard, and then Dr. Zoran Boskovic, and then Jocelyn, I can't pronounce your last name, I can't even read it. That's Widagodu. So it's slightly different order to what you've got in your program, but we go from axon to spine to gene transcription. Okay. Now, have we got any questions to start off with, or should we just launch straight in? Launch straight in. Okay. So Massimo Hilliard has been a long-term member of QBI, and his claim to fame really is that he's very passionate about worms, and he'll tell you why.
Can you all hear me? Good, thank you. First, thank you, Jürgen, for organizing this symposium. This is really an occasion for us to interact with the public and share our passion for things that we want to discover. You might wonder, what the worms have to do with dementia, with Alzheimer. And I hope that by the end of this talk, it will be clear why this tiny little creature might hold secrets to solve some of the pathologies of the brain. And so, and I'm actually happy, I didn't know Lizzie was introducing so nicely this session, which kind of allowed me to go a little bit faster in some aspect. But just to summarize, there is something is not showing you. Our brain is made by 100 billion neurons. And as Lizzie has shown, they need to talk to each other and interact constantly to be able for us to function properly. Now, if we go back to the neuronal structure, this is how a neuron, just in the opposite direction, looks like. And one of the probabilities of the neuron is that they use oxygen to function. So when we breathe, at the same time, the cell themselves use what is called a cellular respiration to be able to function. And unfortunately, a byproduct of that respiration is the formation of these reactive oxygen species. And one of them, for example, is hydrogen peroxide. You might be familiar with it because maybe you got it in the pharmacy sometimes to use as an antiseptic. And it's very reactive. In fact, it kills bacteria, probably kills also some of the skin cells, but it's advantageous because it functions. And this causes damage within the neuron. And unfortunately, these reactive oxygen species, they are a danger because they damage DNA, they damage protein, and they damage the membrane, which is indicated here. This is a lipid fat, a double layer of fat, which constitute the membrane of the neuron. And what the neuron has to do is to deal with this balance between, of course, using oxygen, and at the same time, get rid of these reactive oxygen species which are damaging. You can think of you need to have a garbage truck that comes and removes these oxygen species so that they do not accumulate within the neuron and damage the neuron. And so keeping that balance, it's really important between the oxygen consumption and this accumulation. And when the balance is altered, when we have more reactive oxygen species and free radicals, then we have cell death and diseases. And if we want to bring this into a broader context of diseases, now something that is in common among many diseases, including Alzheimer, Parkinson's, motor neuron, and aging itself, is the accumulation of this free radical and the damage they do to the protein. And so perhaps the accumulation and the aggregation of the toxic molecule is caused by the fact that this damage interacts with the proper function of the proteins. And so it would be really ideal if we could understand and block oxidative damage in neuron and perhaps halt or prevent the generation of the neuron. So the goal is to identify genes that have this protective effect on neurons. But we have a problem. To be able to study a process in detail, we need to be able to control it. So imagine if we had a system where we can induce oxidative damage on demand within neurons. That would be ideal, right? Because now we can precisely control it and understand it. So we want to know when, we want to be able to control when and where that process happens. So what is the solution? It's a combination of two elements. One is a particular protein called killer red that was developed in 2016 by the Lukyanov group in Russia. And what killer red does is a protein that when it's illuminated with the red light, it generates massive amount of reactive oxygen species, okay? So imagine you have now a particular molecule that can be activated, and now there is reactive oxygen species generated everywhere where that protein is located. And the second component is the nematode worm Sinorhabditis elegans, or C. elegans, which is indicated here. This is a common roundworm that probably is in every one of your garden. It's the size of a comma of the booklets that you have in your hand, so it's very tiny, 1 millimeter of length, 50 micron of diameter. And it's very useful because it's a very tractable genetic system. We can do a lot of manipulation with this animal. It's transparent, and in this particular case, it moves sideways. It's almost like a snake, but moving sideways. And we can place this killer red in a subset of the C. elegans neurons. For example, in this case, is highlighted a mechanosensory neuron, which allows the animal to detect mechanical simulation. If the animal is moving and touches something, it's going to back up because it might detect that as a repellent, something bad for it. Now, if we place this molecule within this neuron, in principle, we can simply shine green light on this animal, and we're going to generate reactive oxygen species selectively in those neurons that are expressing killer red, right? And this is exactly what we do. So our experimental system is a population of animals in which there is killer red, the molecule, selectively in these neurons. We can irradiate with green light. This is under a normal microscope. We just shine green light on them. And now these neurons become damaged. So in this case, you can see in this scheme, the neurons completely degenerates and fragmented. However, interestingly, when we expose a population of animals under these conditions, about 60% of them will present this particular degeneration, but 40% of the animal are actually okay. So what this tells us is that perhaps these animals have something that is within their body that is allowing them to be protected from this reactive oxygen species. So it's almost like we're hypothesizing that the neuron have something inside that is protecting them. And if this is true, we should be able to detect what that is. What molecule do they have that is protecting them? That would be a quite interesting discovery. And so so if you want to represent this particular, if we want to represent this particular scenario with 60% of neuron dying and 14% being intact, we can represent it in a bar graph like this, okay? Wild type means these are normal animals, the one that you just seen. And when they are irradiated, about 60% present degeneration, whereas these other here that are not represented are fine. So I was very lucky to have in the lab a PhD, a very talented PhD student, Alessandro Donato, who is here in the audience, who decided to embrace this project and try to find what was the protective mechanism that this neuron had, if they had one. So ideally, what she was looking for is animal in which they've lost this protection, that now suddenly all the neurons die. But how do we do this? So we're basically looking into a large number of animals that have been mutagenized. So we randomly shoot within their genome and looking for those animals in which that particular gene that was protective has now been mutated. It's almost like now you can imagine you have a population of animals that are normal, but now we found a patient in which now that protective neurons has lost the capacity to be protective. And so Alessandra looked to over 10,000 animals in her screen, and she found a particular strain here where now every time she radiates with green light, most of the neuron dies. So this was very exciting for us because it means that the hypothesis that we had was true. So there is something in these animals that is protective. So if we can identify what that is, that would be really interesting because it means that we can manipulate that particular gene and perhaps protecting neurons. And so it turns out that Alessandra, with a lot of genetic analysis and sequencing, was able to find what that particular gene was. And this particular gene is here called, we call it BD60. There's a lot of patent issue going around, so this is the name that was isolated, is a transmembrane molecule. So this particular molecule sits on the membrane on the neuron, and here the membrane represented with this little red and green soldier upside down. This is like basically molecules of fat that form the membrane of the neuron. And the molecule sits right here. And so if this was really a protective molecule, if we provide the neurons with more of it, we should be able to protect it, right? So if we, instead of having one, we overexpress this particular molecule in the neuron, we should be able to have a more protective effect. And this is exactly what happens. Now, the neuron in which we have expressed more of that molecule is able to resist even better than before the stress provided by the active oxygen species. And so we're now working we have taken the same molecule and we have expressed it in different sets of neurons, and we saw a strong protective effect, and we're now putting into vertebrate system to see if we can now extend those results also to mammalian systems. And with this one, we'll stop. I want to thank Alessandro Donato, who is the leading author of this project, but also Eva, who contributed to the screen, and also other people in the lab, people here at the QBI, our support group, also the CGC and consortium providing strains. Rowan Tweedle, she's not here. She's our scientific writer who really helped us to get grants, get funded, our funding agency. And most importantly, I want to thank not only QBI, but also the CJ Kader and Jürgen in particular to make such an amazing place to work and making discovery. It's very cohesive, and probably this will come across now with the following talks, but it's not really a fervent and really nice environment to work with. And with this one, I will stop and I'll be happy to take any questions. Thank you.
So do people have questions for Massimo? There's one here. Down.
About that gene that is taken from C. elegans is then given through mitochondria, or is that the way it works?
So I'll repeat the question. So the question, it's very interesting, is this gene going through mitochondria? Because the mitochondria are the organelles that are necessary within the cell for the respiration. So that's a very important question. We don't think it's there because it seems to be on the membrane. And what we think is doing, and I didn't want to get into detail, what we think is doing is that particular molecule that you've seen here, I'm just going to go back quickly. What we think is doing is allowing the garbage truck that is inside the neuron to be, is able to recruit the garbage truck right in this place near the membrane when it's needed. And the garbage truck is the proteasome, which is a multi-protein complex that function in neuron to clean all the protein that are conformed differently.
How do you administer that genetics of that elegans into, if it doesn't go through mitochondria, where it is, how do you administer it in to give protection to the cell?
Right, so we can, so the question is how we can administer that to the cell. Yeah. In this particular case, there's a genetic way when we can express selectively that molecule within the organism. But the way that we are going now in mammalian system, we are expressing in cultured cells, in cultured neurons, and then treat those cultured neurons with oxidative stress, with, for example, hydrogen peroxide, and see if that particular molecule can protect those neurons from stress.
What's the purpose of the microglia? That's an immune cell which is also responsible for inflammation.
So the question is, you're asking if this one will function in glia.
Yeah.
So we don't know that. What we know, though, is that this particular molecule is sufficient and necessary within the neuron. So to make a very complex the concept simple, if we have an animal that is lacking that molecule everywhere, and we bring it back exclusively within the neuron, we still have a protective effect. So we think that the function within the neuron is sufficient to recover the phenotype.
So that reduces the potential for that oxidation that causes the build-up, promotes the build-up of the beta-amyloid plaque.
Yeah, we did not test specifically on beta-amyloid. We hope that it does, but we did not test if it can protect from beta-amyloid specifically.
In all the diagrams we've seen out, there's been nothing about a vascular dementia.
No, as Massimo said, that there are many dementias where reactive oxygen species is active, but this is very much fundamental. It's not talking about vascular dementia at the moment, but I will just ask, as you said at the end, you're talking, you've found this discovery, which is very, very exciting, that there's a molecule that exists in worms that can stop reactive oxygen species toxicity. So you're now testing it in mammalian cells. Do you know if there is a homologue in mammalian cells, or would this be something that's unique to worms that we could then develop to treat mammals?
At the moment, at the moment there is no human homologue, so we're really hopeful that we can actually push that into the cells and make them function better.
Great. Thank you very much.
Thank you.
So he did his PhD only a couple of years ago, and now he's doing research in one of the labs in Pt2, and he's going to talk to us again about degeneration, but maybe how the brain can try and help fix itself.
Excellent. Alrighty, well, good afternoon. Thank you, Lizzie, very much for that kind introduction, and thank you for the organising committee for inviting me to speak to you here today. And as Lizzie briefly mentioned, really today I'm here to talk to you a bit and give you a bit of a general overview of some of the work that we do in the Puzzle Laboratory, where we try and we're trying to understand and uncover the brain's response to neuronal damage. So obviously there's many ways, when I talk about damage, there's many ways this can happen, and we already heard a great talk by Professor Hilliard about the type of what reactive oxygen species can actually do to a neuron. But really, in our particular lab, we sort of try and apply it to Alzheimer's disease. And we heard in the morning some fantastic work going on in the GOTS lab about trying to understand the effect of the plaques and tangles have on the neuron. But for the purposes of this talk, really it's important to understand that through many various mechanisms and through many processes, this initial formation of these pathological plaques and tangles can lead from a healthy brain to degenerate and lose a lot of neurons and have some pretty devastating cognitive effects, as you can see here on the right. Now, the reality is that this process, depending on many, many factors, can take months, years, and in some people even decades. As you've already heard Lizzie mention that some people have these amyloid plaques for many decades before any cognitive symptoms occur. And for us, that's really interesting. And to explain why, I just want to point this out from a slightly different perspective and really looking at two main factors. One is the passage of time here from left to right, and the other one is brain health. So healthy brain would be, let's say, up here. And if that brain health falls down under a certain threshold, we talk about these cognitive symptoms. Some of the typical things you think about with people with Alzheimer's, we're talking about severe memory impairment, navigation, and so forth. So if we think about a normal brain, over time, there's just this very normal but very, very gradual decrease in that brain health. And however, there is good news about that a lot of research here shows, and as we heard from Pat Walsh as well in the morning, that exercise and intellectual stimulation are key to flatlining this. But in general, there's this slight decrease over time. Now, if you're unfortunate enough that for whatever reason, whether it's genetically or environmentally, you start developing these plaques and tangles at a certain point in your life, then there's this slow but clearly a more severe decrease in brain health. And then eventually at some point, this reaches a point of no return where you start getting these cognitive symptoms and things start to go down south relatively fast. Now, what we are particularly interested in our laboratory is this time here. Okay, after these plaques and tangles are developed, what is it about the brain that allows it to stay healthy and for some patients to not show any symptoms until things get really bad? So there is something there without our intervention that allows the brain to cope with some of these insults that are happening with amyloid plaques, with the tau, with the oxidative stress, whatever it is. Something is going on there that allows the brain to cope with this. So Lizzie already introduced the neurons, so I'm not going to spend too much time on this. And the key point that I would like to make is that really, when I talk about the neuron, there's this signal direction, and the communication always goes here from the dendrites through the soma and then gets passed on to the axons and the following cells. And just quickly here, this is that same graph we showed before. Now, so this is obviously a very simplified diagram. I'm showing you here one neuron connected to other neurons, but as Massimo said, we're talking about 100 billion cells here that are connected to about a trillion different, trillion different connections to other cells. So we really think that in order to understand what is happening in the brain, the key relies at these individual points of contact that I've just outlined here. So if we just zoom into that little area, and this is obviously a cartoon representation of that, this is what it would look like. So here we have the incoming cell with the axon, and this is our receiving, our red cell that was here. And on our receiving end, we have these small mushroom-like structures that are actually called spines, or in this case, dendritic spines. And we think that these are the key to understanding why the neuron is able to compensate for this damage. Now, obviously this is a cartoon, but what I'm showing you here now, this is an image that's taken actually from our lab. And so this is one stretch of dendrite, and we can see that there's just this small stretch here has about 30 of these mushroom-like structures that are called dendritic spines shown here. In reality, a single neuron can have up to 1,000 of these. And so what that means is that you have 1,000 incoming signals into a single cell, and then this all has to be processed into a unified message that needs to be passed on. So you can imagine the level of control that is required to achieve this. So really, we have many reasons to believe they're key. This is not just us theorizing, but there is a whole group range of diseases that are called spinopathologies. And these diseases, at the very earliest stage, before even the occurrence of any cognitive symptoms, show us severe loss in these dendritic spines. These diseases include schizophrenia, autism spectrum disorders, Parkinson's, Huntington's, and of course, relevant to us here, Alzheimer's disease. Now, despite you may know some of these other diseases for many other symptoms, but the reality of the situation is that all these are underlined by severe cognitive symptoms. And what you can see here, this is a comparison between just a very, very late stage Alzheimer's dendrite and a normal dendrite. You can see the amount of loss in spines that happens. And the amount of spines that are lost are directly proportional to the cognitive deficit. So that tells you how important these structures are to providing normal neuronal communication within the brain. So the question that we're asking, is there a way for the neuron to recover from this damage? Now, back about 30 years ago now, there was this paper that came out, and just to summarize everything, what these people looked at post-mortem Alzheimer's brains, and what they show that even though in these brains there was about a 30% overall loss in these spines, what they saw is that the remainder of the spines grew larger. And we found that fascinating. So why is that important? So if I bring up that previous picture that I showed you here, you can clearly see that some spines are smaller, whereas some spines are larger. And the size of this spine directly corresponds to how strong the communication at that point is. So we think that something about the spine enlargement is a way of the neuron to respond and try to restore some of that lost communication that happens with neuronal damage. So for us to test whether spine enlargement is some sort of neuronal compensation, we do a very similar thing to what Professor Hill already explained, where we try to artificially induce this in a controlled environment to then observe what are the effects on some of the surviving spines. And so just to briefly give you a very, very short overview of what we're finding, is that if we take a picture of a neuron and then on day one, and then 24 hours later after we've eliminated some spines, and you can quite see here there's a severe loss of spines. I've indicated here that some of the remaining spines get quite a lot larger, and this is what we show here. Now, what's the interesting part about this is this that I'm showing you is actually found in young neurons. However, we have some very early and exciting data that this ability seems to be lost as the neuron gets older. So there's something about the aging processes itself that prevents this from happening. Now, the encouraging thing is that if we look back on some of these younger neurons, what actually happens is that when we do our elimination, you see there's a spine loss here, and I've indicated quite a few of them, and you still see some of this enlargement in the surviving spines, like I'm showing a couple of examples here. Over time, some of the spines that have been lost actually grow back. So that tells us, is this initial enlargement the type of defense mechanism until the neuron can at least attempt to recover some of these lost spines. So we'd just like to summarize all of this for you and really give you an overview and a plan of where we're trying to do, what we're trying to do here. So as I told you, typically what happens is you get these plaques and tangles and you get this overall decrease in brain health. And we think that somewhere along the way, most likely very early on in the process, there's an overall loss of spines. However, I think when that does happen, the brain's first response is to try and compensate for it. Okay. Now, the issue here is that what we're doing here is in a controlled environment, and we've heard today the many, many aspects that get disrupted in Alzheimer's disease. So it's not just about a loss of spines. Eventually, the cells die, there's reactive oxygen everywhere, mitochondria, all these things get affected. And the problem is that after a certain point, this is not enough. And we think that's why it's very hard to stop this with the innate defense mechanisms. But what we think is that if we can understand this process, maybe there's a window of opportunity there for us to use these natural processes to actually boost this defense mechanism and stop, or in an ideal world, try and recover some of these lost cognitive symptoms. So I think with that, I'm pretty much on time. So I would just quickly like to give a thanks to the Pazzo Laboratory and Dr. Patricio Pazzo, and this project is really his brainchild. And of course, the other members of the laboratory, and especially thank you for your attention.
So as you can see, we start with Massimo, who's already found a defence mechanism and isolated that, and here looking for another naturally occurring defence mechanism that may be able to be developed. So have we got any questions for Zoran? No, anything pressing?
Okay.
No? Yeah.
Yes.
In all of this, does what you eat and how you live impact on all of this?
Yes, of course.
It has to impact.
Yes. Well, it's hard to nail a percentage, but we have some leading experts here that are working on the importance of exercise and intellectual stimulation, of stopping this natural aging process. And of course, diet is a huge factor of this. And so there's always a balance between the environment and the genetics. So the more things you can do in your life, diet, exercise, keeping intellectually active, obviously those things are all going to help balance out if you're unfortunate enough to have some genetic predisposition. But the whole thing is always a balancing act between nature and nurture.
And we don't know that eating broccoli is particularly good for keeping your spines alive or anything like that. But we do know that cardiovascular health and exercise and maybe the blueberries that mop up reactive oxygen species are good, but we don't know. Yeah. So thank you, Zoran.
Thank you.
And we'll move on to our next speaker, who's I don't know if that's an official title, but it's basically a sort of a junior faculty. She's running an independent project, but is not yet a professor, but I'm sure she will. So our next speaker is Jocelyn, and she's going to talk about the changes that happen in our genes when we've got Alzheimer's disease.
I just like, is this on? Just turn it on. Yeah. All right, yeah. So thank you for staying back till this final part of the session. So I'd just like to start with my first slide, and this has a lot to do with the latest questions about how our environmental stimuli can actually affect our body's response. And this is the nature and nurture in play. So we all agree that genetics play a big part of who we are. On the right here is a photo of my 4-year-old daughter. And some of you might agree that she doesn't quite look like her mother, but thankfully she looks a lot like her father. And in addition, she has her own personality and quality, which neither of us do. She loves singing and performing in front of people at a very young age. And during her development, she will constantly be exposed to learning and life experience, which continually shapes her life, her character, but also her DNA profile. By this, I don't mean changes in her genetic code, but rather changes at the DNA epigenetic level. So epigenetic mechanisms plays a huge part in the nature and nurture way of shaping our lives. This research on epigenetics was highly investigated in the year in early 2000, and for over a decade the research intensifies. And in 2010, this image of cover from Times magazine actually provides these highlights, and it's one of my favorite headlines that says, "Why your DNA isn't your destiny." So epigenetic changes occur throughout life. We've heard from this morning that there are many hallmarks in the aging process, and epigenetic control is actually one of the 9 hallmarks that's in Jürgen's diagram. In the past 5 years, research have shown that normal process of aging is accompanied by changes in our epigenome, which scientists postulate as the epigenetic clock that runs vertically, so that based on their epigenetic changes, one can actually predict the chronological age of a person. However, we know that many factors can influence the aging processes. These include our diet, physical exercise, or stress level. So environmental factors can influence whether the clock is ticking faster in the case of premature aging or disease state, or whether the clock is decelerated in case of healthy aging. So how does this epigenetic mechanism work? So I'm a molecular biologist, and I'm interested in how things work at the molecular levels. And just to reiterate some of the things that Lizzie has introduced, in each of our cells, including neurons, there is a flow of information from DNA, RNA to protein. And the same DNA or genetic code is actually shared by each individual cells in our body, whether it's a skin, muscle, or nerve cells. However, through a process of transcription, this is where genes are being expressed. They're copied into this messenger RNA molecule, which then translated into proteins, which is also known as the building blocks of life, as they form various complex molecules that define what cells does. So epigenetic mechanisms can tune the level of gene transcription through various mechanisms. Now, the best studied mechanism is by adding chemical tag on the DNA. So rather than changing the base that is already encoded in this double helix structure as we recognize what DNA is, it actually adds a chemical tag, or what we can think of as switch on the DNA. Now, the other mechanism is by affecting this histone core protein that looks like a circular ball where DNA is wrapped around with. So these mechanisms can influence strongly how our genes are expressed in the body. Now, my current research is actually on the next level, which is similar to epigenetics, but it's actually at the RNA interface level, or what we know as the transcriptomic layer, because it's the product of transcription process. And this is also known as the epitranscriptomics, which is above transcriptome regulation. Now, this is a very recently emerging field that is very much similar to epigenetic control of DNA, but targets this single-stranded RNA molecule rather than the double-stranded DNA. So the study of RNA has its own challenge for a long time that hamper its progression, and not until recently, with improved sensitivity of sequencing technology and analytical tools, we learned that there are over 100 of these distinct types of chemical tags that actually can be deposited on RNA. So these chemical tags have the capacity to determine the lifetime, the characteristics, or the rate of translation of RNA molecule into protein. And one of the most abundant type that has been characterized today is this particular modification known as the M6A. So this is a specific methylation on the adenosine residue that was initially actually reported back in 1970s, but not until recently in 2011 and 12, where the research has actually taken off. And this is because sequencing technology has made scientists realize that this modification is very abundant and pervasive in mouse and human cells. It's also important to note that most of these modifications are reversible. So there are enzymes that are actually responsible for depositing it, and also enzymes responsible for removing it, making it a very dynamic process. And having characterized some of these enzymes, researchers can actually make use of this enzyme to manipulate the level of M6A or other modifications in the cells and look at what is the biological importance of these RNA modifications. One of our long-term interests in the lab is to understand how basic learning and memory processes is regulated at the molecular level. So a few years ago, we published this paper that provides the first evidence how M6A is important in the learning and memory process. And this was done by targeting an enzyme that is important in the biogenesis of M6A, known as FTO here. So if we actually target this enzyme in the prefrontal cortex region of the mouse, we actually boost up the level of fear memory processing in these mice, in this fear-related paradigm. Now, several papers were published last year by various groups, further highlighting the role of M6A in learning and memory. And given this importance, the lines of evidence of M6A in cognitive function, we speculate that M6A dysfunction may contribute to the onset of cognitive decline in dementia, such as Alzheimer's disease. Now, it's important to note that we don't have any prior knowledge of how M6A behaves in normal process of aging. So this makes up our main research goal, that is to characterize how M6A profiles actually shapes, how M6A profile is shaped during normal aging as well as pathological aging, such as Alzheimer's disease. So this is a new investigation in the lab, which I'm quite pleased to share with you some of our exciting preliminary data that indicates the involvement of M6A. So we started by examining the existing database of gene expression and zoom into each of these molecules which are responsible for M6A control. So shown here is a huge complex that is responsible for depositing M6A. Now, our analysis found that metal-3, which is marked here, which is also a key enzyme of M6A deposition, is actually downregulated in the hippocampus region of dementia cohort. And hippocampus is, as you've heard from this morning, is where memories are formed and stored, and this part of brain region is severely affected in Alzheimer's disease patients. And a similar pattern also is shown in the prefrontal cortex and also in the temporal cortex of the AD patients. Now, we were lucky to have also some tissue samples from our collaborator, so we can directly test this in the lab. And indeed, we found that there's a consistent downregulation of these enzymes in the AD samples. In addition, we've also heard a lot about aggregation in neurodegeneration, where protein normally aggregates in insoluble fractions. So we've used insoluble fraction to look at how this protein behaves. Now, similar to aggregation of the tau protein, we saw that Pt2, which is the enzyme for M6A, is also enriched in this insoluble fraction. So this early evidence suggests that abnormal expression of Pt2 suggests a potential dysfunction of the M6A epitranscriptomics in Alzheimer's disease brain. So the current work that we're doing is to look at, is to map out the location of M6A throughout the transcriptome during normal aging by comparing a young mice and old mice. So these are 3 months old mice compared to a very old 22 months old mice. And we would like to find out how this M6A signature is different to a well-characterized AD mouse model, such as an APP-PS1 mouse, which harbor the human mutations, human AD mutations. So we do this by isolating RNA from the hippocampus and then perform an enrichment process, followed by a high throughput sequencing, which allow us then to quantify the relative levels of M6A throughout the transcriptomes, and this can be anywhere between 4,000 to 7,000 genes. And then finally, by intersecting the profile of the normal aging and the AD mouse profile, we hope to define what is normal M6A profile and what is unique to the AD M6A brain profile. And in near future, we'd like to extend this to human cellular models using iPSC's model system, whereby we can actually isolate nerve cells from fibroblasts, isolated from AD patients, and then perform the similar experiments to look at the M6A profile through a sequencing approaches. And this is particularly important because we know that majority of the AD patients are sporadic. So unlike our mouse models, where we have a predetermined genetic basis, we can actually look at this in the sporadic AD case. So in summary, we have M6A epitranscriptomic that exists in addition to epigenetics that is crucial for memory and cognitive function. Now, this is a very new area of investigation, and we do not know how this molecular regulation works during normal aging versus pathological aging. So this is what the major aim in the lab. Now, we've got a preliminary data that shows that changes in the level of M6A enzyme in AD hippocampus that suggests for a potential dysfunction in this RNA signaling, and therefore characterization of this RNA regulation in neurodegeneration might hold promise for novel biomarkers and potential therapeutic target. And just as a last note, I'd like to also note that epitranscriptomics is currently heavily investigated in the cancer research field, and it's certainly one of the major target and highlighted this year only. So just to thank where this work is being done in Victor Angona's lab, and this project is a major focus of our PhD candidate, Ms. River, and our samples has been kindly shared by our collaborator, Dr. Rodrigo Medeiros, and the UCI MIND Institute. Thank you very much for your attention.
So thank you, Jocelyn. It's a real tour de force of molecular RNA and DNA, and it's just really highlights that QBI and CADRA are really pushing the envelope of what's possible to look at, as well as the fantastic techniques that Matsumo and Zoran have described to you. So have you got questions for Jocelyn? So maybe just to kick things off. So at the moment, you know that there are changes in one of the molecules that form the complex that changes the methylation. Do you know whether anything about the targets of the RNA that get modified in this way? Is it lots and lots of different targets or just one or two?
That's currently the aim of our studies, starting from mouse models. So we're looking at unbiased approach. We'd like to sequence the whole transcriptome and find out where this methylation mark is deposited.
And so what that means is if you can see which RNAs are being marked and therefore maybe being regulated up or down, then if that molecule isn't active in aging or in Alzheimer's disease, then those proteins that are going to be needed for the normal function in the nerve cells are also going to be up or down regulated, and therefore maybe make the cell less able to cope with what's going on in terms of amyloid or reacted oxygen species, or not being able to grow its spines out. So this is very fundamental research to just understand the normal aging and dementia profiles of what's going on in our cells. Yeah, Jürgen.
Because we have so many somatic mutations in human brains as we age, and particularly also in neurodegeneration, are you planning to map the somatic mutations in DNA in the brain against RNA changes?
At the DNA levels, that's not really focus of our study, but the DNA versus RNA. No. Because the RNA itself is complicated enough. But certainly, I mean, we'll do a lot of comprehensive cross studies with what's published in the literature.
So what Jürgen was alluding to is, because our nerve cells are with us when we're born, that over time, each individual cell, the DNA may change, and so you might have one particular mutation within one cell. Now, this is something that happens in cancer, and if that changes in a bad gene, then the cell proliferates and you can get a cancer. But in Alzheimer's, that might trigger the nerve to degenerate or to produce something toxic, beta amyloid or so forth. So trying to tie those changes in individual cells with individual RNA changes, I mean, yeah.
It would be nice. It would be nice to have everything.
Great. Well, that's, unless if there's, one more question up the back.
Sorry, I'm a layman to all the people here, because I'm an artist, that's my background. But my father has dementia, so that's my interest, and that's what brought me here. But what I'm interested in as well is the effects of things, and people have talked about free radicals, things like pesticides and how they actually affect the cells and the neurons, and is that really being looked into?
Yeah, I'll answer, and then maybe Massimo might have something to add. So in terms of dementia, as far as I'm aware, there's not a strong link between dementia and environmental toxins such as pesticides or metals. There was a theory for a while about aluminium pots, which was not correct. But that doesn't mean exposure doesn't tilt the balance a little bit. But Massimo, do you have anything?
Yeah, so there is a very strong link between paraquat, which has been used as a pesticide, and Parkinson's disease. So there is definitely a link there, and that causes oxidative stress in neurons. So in fact, that is used as one of the substance to cause neuronal death and also aging. So there is definitely a link there, although I'm not aware of specific with Alzheimer's disease, but definitely with other diseases.
Okay, well, thanks guys, well done. And I'll now hand back to Professor Gotz to close the session.
First of all, thanks a lot, Lizzie. You have done a great job in basically paving the way and integrating all the excellent work which is going on. Victoria and Katarina prepared these notes for me, thank you notes, but obviously we didn't include their names, but it's my absolute great pleasure to thank Victoria Anderson is here, Katarina Ellert, I've seen her just a second ago. Oh, there she is. They have done such a fabulous job in organizing this and making this such a great event. They do this, I mean, this is an event which is now happening already the 5th time. So thank you very much. And let's give them a special applause. I would also like to thank Evelyn Moore, first exposure to her in both ways, I would say. Thank you very much for guiding us so expertly through the time. I would like to thank all speakers of the session, and I would like to thank particularly you who come here on a regular basis, also for those who come for the first time. It makes such a difference for us to see your support and your enthusiasm and your encouragement, because that's what we need when we stand in the lab. So thanks a lot, and see you again next year.